Dear ENS satellite meeting participants, hello, my name's Nicolas Chevalier. I work at CNRS at the University of Paris. I'm very honored to be here. I would like to thank the organizers, and we'll be talking about the embryogenesis of gut motility. As you all know, the gut is a complex system with many different cellular types, and motility will depend on many different inputs, both mechanical and chemical, giving rise to a variety of different motor patterns and many different functions. So our idea was that by looking at the embryo, we could figure out what the basic nuts and bolts of motility are, how they assemble together to give rise to adult motility, and uh, and this way get an understanding of the fundamentals of motility. Also try to get some insights into pediatric motility disorders, and finally see how motility in the embryo may affect other developmental processes such as growth or differentiation. Other species which have been considered in the development of motility include the zebrafish, the mouse, with many awesome studies by you investigators in Australia. And over the past couple years, we have performed studies on the chicken, and I'll be giving you insight into our discoveries today. So the first movements in the chicken start at 6 days of development. So as soon as a ring of circular smooth muscle differentiates inside the gut mesenchyme, you'll see motion like this. That's an accelerated movie. So you see waves propagating at constant velocity, left and right, nucleating in some points. When they meet, they'll annihilate. So this movement is purely myogenic. If we apply tetrodotoxin, it does not have any effect. And ICCs are not yet involved because they're not yet differentiated, or they are not yet active. I'll come back to this later. We were interested in imaging the basic electrochemical wave which underpins this early motion. So we performed calcium imaging using fluorophore AM, but we needed it to get inside the smooth muscle. So our technique was to embed the gut in agarose, perform thick slices around 100 micrometer, and believe it or not, we still get motility. That's what it looks like. We get an inside view of these lumen occluding contractions. And now if we apply fluorophore AM, we find streaks of light flashes that propagate circumferentially and accompany each contraction. Recently, a group led by Taben, the first author is Huike, presented some beautiful videos by electroporating a G-CAMP reporter of the same phenomenon. You'll see the contractions as well as the underlying calcium wave. So we figured out some basic properties of these early calcium waves. The fluorescent spikes have the typical shape of calcium spike in smooth muscle with a sharp uprise and slower decrease. Sometimes we get two calcium spikes for one contraction. Sometimes we get calcium spikes but no contractions. We found that the calcium wave travels through gap junctions. If we inhibit them with enoxalan, the calcium waves, the contractile waves will eventually vanish. We were then further interested in knowing whether there could be any mechanical component into the that played a role in how the wave propagated from cell to cell. Would cell tension play a role in propagating this wave, a bit like in a violin string? So we performed an experiment where we would stretch the gut by some amount, up to 50%. You can see this here in the graphs, and see whether there was any influence or on speed or frequency. We could not find any. In another experiment, we would block a piece of a small region along the gut by embedding it in a stiff agarose gel and trying to see whether waves would either be absorbed or reflect or be transmitted. And you can distinctly see here on the motility on the space-time diagram that they go from one end and are transmitted to the other. So our conclusion is that mechanics does not play a role in wave propagation. The phenomenon seems to be purely electrochemical. But wave initiation will be mechanosensitive, and this we could prove by a very simple experiment. We press the gut locally by a pipette, and you see as soon as we press, we get two waves that are outgoing left and right from the point where pressure was applied. This reflex is even sensitive to very light stimulus. A faint brush is enough to trigger it. It's not neurally mediated, and this is all very reminiscent of the pressure-induced reflex. I'll come back on this aspect later in the presentation. So now, what are all these waves good for? Why does the embryo spend so much energy on propagating waves that are basically only transporting amniotic fluid? Um, in the lab, we're actually able to grow the embryonic guts. That's what we see on this picture. You have an E10 gut initially, and after 2 days in culture, when we supply enough oxygen, we get a significant increase in length of the gut. And we could show that this elongation depends on the contractions. If we inhibit them with nicardipine, we abrogate lengthening and instead get a diameter increase. So it's still growth, but by getting fatter in a way. Similarly, if we cut the line of muscle tension by cutting one of the gut walls, we still get growth, weight increase, volume increase, but no lengthening, it's thickening. So we understand that by considering that the embryonic gut has a very small lumen. So actually when circular contractions compress the gut, they will cause a Poisson effect, which will cause a longitudinal strain component, and that incrementally causes elongational growth, which has recently been shown to be mediated by the protein YAP. So I'll move on to step 2, rhythmicity. We noticed that there was a very different spatiotemporal map between E12 and E14. Waves would be bidirectional and somewhat irregular at E12, and as from E14 they become very regular clock-like. So what is this due to? And what is this sharp speed increase due to? We guessed that ICCs could be involved in this behavior, so we labeled them using in situ hybridization for KIT and for TMEM16A. We found first KIT expression as from E10 in the chicken embryo, for TMEM16A as from E7, and if we look at the morphology of the ICCs, that's what we find. It's a very fine mesh network, similar to that of the mouse. By somewhat modifying our fluorophore AM labeling protocol, we could image calcium activity in Cajal cells. That's what a typical movie of this activity looks like. So there are calcium flashes in non-contractile cells, which we have found to be the Cajal cells. So it's very regular and only occurs as from E14, which is in agreement with the changes in the motility diagrams I just showed before. So now what if we inhibit the Cajal cells in this critical transition from E12 to E14? We used imatinib. It causes the ICC network to atrophy, as had been found also by Beckett in a previous publication. And if we look at the spatiotemporal maps now for guts at E12 plus 3 days in culture, in the control we get these very regular contractions, but in the imatinib treated samples, the contractions are chaotic, very irregular, reminiscent of what can be observed in some diabetic rats or CIPO spatiotemporal maps. So now let's move on to step 3, which is the orthogonal muscle layer. Longitudinal muscle will start differentiating in the chicken embryo as from E14. We can see it here distinctly on this IHC at the outer periphery. That's what the contractions look like as from E14 on the longitudinal axis. There is this jitter motion as to the circular contractions. We can actually differentiate them optically on whole mounts because the longitudinal contractions will give rise to a sideways buckling of the sample. So if we look at the average position, we recover the longitudinal contraction frequency of about 0.3 CPM, whereas if we look at the pinching, we find this high frequency signal at 3 CPM, which are the circular contractions. So we were interested in knowing whether the two are correlated. Is there any coupling? We found none at E14, but at E16, we could distinctly observe that when longitudinal muscle was contracted, there were no more circular contractions, or their amplitude was strongly diminished. So how does this occur? That's where the enteric nervous system comes into play. We found that it first becomes active at E16. That's the first reaction to tetrodotoxin. That's what this network looks like by Tuge immunohistochemistry and a 3D transporized duodenum. To summarize our findings, we found that this first activity is inhibitory. It is mediated by nitric oxide. We think that the mechanosensitive coupling between the two muscle layers is the following. When longitudinal muscle contracts, it will compress the ganglia, causing them to release NO, which relaxes the circular muscle layer. And this coupling gives rise to a new motor pattern, which you'll see in this movie. So first, you'll see high frequency circular waves and propagating from left to right, this migrating motor complex. There it is. Again, again. So this is the first polarized motor pattern which we could observe in the embryo. And as we see it, we can define it as a rostrocaudally propagating longitudinal contraction, which is neurogenic because it is abrogated by tetrodotoxin, as you can see in this spatiotemporal map. Now, what about the reaction to mechanical stimulation? What if we pinch an E16 gut? That's what you'll see in this video. We first have circular contraction waves. We pinch here, and aborally from the point where we pinched, we have an inhibition of circular contractile waves, which we can see distinctly on the spatiotemporal maps as well. If we inhibit neurons with tetrodotoxin, we recover the earlier myogenic response where we have two waves outgoing from the point where we pinched. So this is important because it basically explains where the famous law of the intestine, ascending contraction, descending relaxation, comes from embryologically. Basically, in the early embryo, we pinch, we get two waves. It's a symmetric reflex. In the late neurogenic embryo, we pinch, the wave going towards the stomach still contracts. There's an ascending contraction, but the wave going aborally is inhibited by neurons. We get the descending relaxation. So to conclude, I've driven you through different phases of motility development in the chicken embryo, going from circular smooth muscle waves, then how they become rhythmic because of the activity of ICCs, how the orthogonal longitudinal muscle layers gives cross-direction contractions, and finally, how these cross-direction contractions are coupled by the enteric nervous system. What's interesting is that this chronology is exactly the same in humans, but not in days, in weeks. Here you can see on this MRI image on an aborted embryo from the Kyoto collection by Ueda. The first contractile waves are seen as undulations along the gut. And it has recently been shown by McCann and colleagues that the first enteric nervous system activity starts at 16 weeks in the human embryo. I'd like to thank all my collaborators, interns that worked on this project, and would finally like to invite you, if you come by Paris, to visit our institute, which has a range of activities from hydrodynamics to biomedical engineering. And with that, I'll be happy to take any questions if this is technically feasible. Thank you.